Good evening, boys. I am Ilangovan, online at c.com personal for teaching zoology. So, in that chapter, we talked about the process of transcription and molecular genetics or molecular biology. And we have concluded about the process of transcription in bacteria. And what are the two important significant characteristics of transcription in bacteria? Unlike transcription in eukaryotic cells, here in bacteria, the mRNA does not require any processing, namely the splicing process or capping process or tailing process or addition of any other structures to the mRNA. So there is no processing required for the mRNA that is being normally transcribed by the DNA during the process of transcription. So it directly becomes active or it becomes a mature mRNA. And again, secondly, the second important character, both the transcription and translation normally takes place in the cytoplasm, not in two different compartments. As a result of these two events, we have many times during the process of translation or during the process of transcription or what we have during that process of gene expression, translation can begin much before the mRNA is fully transcribed. So the translation and transcription takes place simultaneously. There is no separate events of transcription and translation, and both occur simultaneously. That's why we can say the prokaryotic translation is normally coupled with the transcription and occurs co-transcriptionally. I use the word co-transcriptionally. The meaning for that one, there is no separate events of transcription and translation, both normally taking place simultaneously in the cytoplasm. There is no separate compartments for transcription and translation. So this is the main thing what we have with reference to transcription and translation in the case of bacteria, one of the important characteristics. Unlike the eukaryotic cells, there you have the mRNA has to undergo a process of what is called the processing. And then we have the splicing process. Then we have capping process, tailing process. All events have to take place to just complete the transcription process. But here all events have not taken place. There is no processing. Directly the mRNA form is active and that one is considered as a mature mRNA. Now let's pass on to the next part of this topic. See, just to compare with the help of diagram, you could see in the case of prokaryotic cells, there is no definite nucleus. We have the chromosomes found in the cytoplasm itself, the DNA. So that's why we have the process of transcription and translation normally taking place simultaneously in the same compartment. But in the case of eukaryotic cells, you see that one, the transcription process takes place within the nucleus because we have a definite nucleus. Then after that one, the mRNA has to undergo a process of what is called splicing, capping, tailing, etc. And that process is called what is known as a processing. After that, it has to exit through what is called the nuclear pores, that is called the transport, and eventually it is being translated. So the transcription process occurs in the nucleus and the translation process occurs in the cytoplasm in two separate compartments. And also we have the process, namely the processing, and that is not taking place in the case of prokaryotic cell. We will discuss in detail about this eukaryotic transcription. Now transcription in eukaryotes. So in the case of eukaryotes, there are two major complexities. When compared to the prokaryotic cell, there are two additional complexities. Complexity number one. See that one, there you have only one RNA polymerase, and that is responsible for the synthesis of all the three types of RNA, tRNA, mRNA, and also rRNA. But in the case of eukaryotic cells, the first complexity is this one. So there are three RNA polymerases, namely RNA polymerase 1, 2, and 3. And they are all essential for the synthesis of the three different types of what is called just the RNAs. So that is why we can say there is a clear-cut label division, or we have a clear-cut division of label. A particular just what is called RNA polymerase enzyme is responsible for the synthesis of a particular specific RNA, either mRNA or ribosomal RNA or tRNA. So altogether we have three different groups of RNA polymerases. One, RNA polymerase 1. Number 2, RNA polymerase 2. And number 3, RNA polymerase 3. Now the RNA polymerase 1 is responsible for transcribing what is called the ribosomal RNA. So the ribosomal RNA normally 45 Swedberg unit, and it is being composed of different units of 28S, 18S and 55S. S stands for Swedberg unit. The sedimentation coefficient constant. So RNA polymerase 1 transcribes what is called ribosomal RNA. Then number 2, RNA polymerase 2, which transcribes what is called the messenger RNA. The messenger RNA is normally produced in an inactive form or just actually pre-mature form or pre-mRNA form or primary transcript without being processed, and that is why it's called as heterogeneous nuclear RNA. Heterogeneous, it is not uniform, having two different components. One, the coding sequence, namely that is the exons, and the non-coding sequence, namely the introns, which are being actually transcribed by the gene. What we can say, the split gene we described already, we have to go through once again. So RNA polymerase 2 is responsible for the synthesis of what is called mRNA. It transcribes mRNA, or also called heterogeneous nuclear RNA, because it is being formed of both wanted and unwanted components which are being translated or which are being actually formed or transcribed. Now the RNA polymerase 3, which transcribes what is called the tRNA, and also a small segment of RNA, namely 5S ribosomal RNA, is also synthesized or transcribed by this polymerase 3. And one more just RNA, which is normally involved in some other events, what is called small nuclear RNAs. So RNA polymerase 1 for ribosomal RNA, then RNA polymerase 2 for messenger RNA, and RNA polymerase 3 for transfer RNA along with some other. So the first formed primary transcript of messenger RNA, as it contains both the sequences of exons and introns, it's also called as heterogeneous nuclear RNA. Before being processed, it is called heterogeneous nuclear RNA. So this is first complexity, that is a clear-cut division of labor, where you have three different types of RNA polymerases to synthesize or to transcribe three different RNAs. Unlike the prokaryotic cells, where you have only one RNA polymerase responsible for transcribing, that is all the three types of RNAs. Now complexity number 2. Complexity number 2. And before going to that one, just I mentioned here a clear illustration, a picture showing that is simply the transcription of what is called the gene responsible for, namely the ribosomal RNA, which is being catalyzed by RNA polymerase 1. So this is the transcription unit in the DNA molecule, so which is being transcribed. So this is what is called actually the transcribed part, having both exons and introns, and that is normally we have, this is one segment which is being enlarged. A segment of DNA considered as a transcription unit which is being transcribed, so it is being enlarged here. Now then transcription with the help of RNA polymerase 1, we can get what is called pre and matured inactive ribosomal RNA, having a size of 45S, having the units 18S, that is 5.8S and also 28S. Then after processing, so what is happening during processing? The unwanted segments, namely that is introns have been removed, the exons are joined together, so that we have mature RNA molecules. So we can have three different types of RNA molecules, that is the units of ribosomal RNA, 18S, 5.S and 28S. These are all the different types of ribosomal RNA based on their sedimentation coefficient. So this is one illustration showing how far RNA polymerase 1 is responsible for the synthesis of different types of ribosomal RNA. There are three types what I mentioned based on their sedimentation coefficient. Now complexity number 2. As I mentioned earlier, So in the case of just actually the primary transcript, the one which is formed immediately as soon as transcription is over, having both exons and introns, that is called primary transcript. Unprocessed transcript mRNA, having both exons and introns, also called heterogeneous nuclear mRNA. So that should undergo what is called the process, a phenomenon, what is called that is processing. So as it contains exons and introns, you have to remove the non-functional unwanted introns from that one. So that pre-mRNA or the primary transcript must undergo a process called splicing. So what is splicing? Splicing is normally taking place in the case of split gene. What is a split gene? We have seen already. A split gene consists of a coding sequence of DNA, what is called exon, and also non-coding sequence of DNA, what are called the introns. So during splicing process, the unwanted non-coding sequence of DNA, namely introns, have been removed and the exons are joined together, and that process is called splicing. Removal of introns and joining of exons to form just what is called mRNA, where exons are arranged in a defined order, in a definite order. So we have also another process what is called alternate splicing. Sometimes an mRNA may be spliced in several different manners. So that what will happen, allowing one gene or a single gene just to encode for multiple proteins. This process is called what is known as alternative splicing. Sometimes what will happen, the pre-mRNA, the unmatured mRNA may be spliced in such a way that it is allowing what is called actually the single gene coding for many proteins. I will show in the picture. Normal splicing, see that one we have, this is an example for alternative splicing. Now this is the gene in DNA having both exons and introns. The yellow spot represents what is called the exon, and now this one is the intron, unwanted elements. So what will happen during that process, they are being transcribed. So what will happen by a process of splicing, now we have removed the introns. Now in this case the introns are removed. This is a normal splice. Then after that what will happen, the remaining exons may join together in several different manners. For example here exon 1, exon 2, exon 3 join together, and which are being translated to form protein A. And in the second case, exon 1, exon 2 and exon 4 instead of 3 join together to form what is called, that is a matured RNA, namely mRNA, then which is being translated to form another protein. So here we have the RNA undergone different process of splicing, so that allowing a single gene, now here the single gene, to encode for many proteins. Here one protein, another protein, protein A, protein B. This is because of the splicing that occurs in several different manners. So normally what will happen, all the exons are joined together to form what is called a matured mRNA. But in this case, the exons are joined in a different manner, so that we are getting matured mRNA of different types. This is matured mRNA, this is also matured mRNA. So the combination may happen in different manner, so that we are getting actually different proteins. Many proteins are being encoded by a single gene. This process is called alternative splicing that occurs in a rare instance. Now, who is the person responsible for the process of splicing? The splicing is normally done by a protein RNA complex, a structure, namely spliceosomes. So spliceosome is nothing but a structure of combination of RNA and a protein, and that brings about the process of removal of unwanted elements, namely the introns, and joining of exons, the wanted elements, to form a mature mRNA. That is by the spliceosome. Sometimes what will happen in some cases, the RNA itself is capable of catalyzing their own what is called the splicing process. Without the help of spliceosomes, some RNA molecules, they have the ability of splicing. They have the ability of catalyzing their own splicing. In such cases, the RNAs are described as ribozymes. So ribozymes are nothing but the RNA molecules which are capable of catalyzing their own splicing process. This is a rare instance. For example, the case of bacteria, one RNA, namely 23S ribosomal RNA, which is acting as an enzyme capable of catalyzing the process of splicing. So in such instances, we can say that RNA molecule playing a catalytic role, hence called ribozymes. An example of enzyme, that is an example of enzyme, it is normally brought about by RNA molecule, the catalytic activity. Now, number 2. So, under the complexity number 2, one is splicing. So, another event that is under the complexity number 2, the heterogeneous nuclear RNA has to undergo a process of processing. That is additional processing in addition to what is called the splicing process. Hence, they are called additional processing, namely the capping process. Another one, the tailing process. Along with addition of, that is 3-UTR. The letters UTR refers to untranslated regions. So in the matured RNA, we have not only exons but also some regions, and which are normally considered as untranslated regions, which are not being translated. Though present in the mature RNA, they are not being used for the translation process, and that is why they are called untranslated region. So we have the addition of untranslated regions both at the 3' end and also at the 5' end. So we have 3-UTR and 5-UTR. And as soon as all these events happened, completed, now the matured RNA, namely the mRNA, just exit through to the cytoplasm through the nuclear pores. So the export of that one is also needed. So these are all the additional events. So what are the additional events you have? Number 1, the splicing process. Number 2, the capping process. Number 3, the tailing process. Number 4, the addition of, that is 3-UTR and 5-UTR, along with the event, namely the exit of the formed mRNA, the active mRNA or the matured mRNA through the pores of nuclear membrane to reach the cytoplasm. Now we have to go, what is capping process? What is its importance? So we have the mRNA being formed, but the capping process also begins as soon as the transcription process starts. Once the transcription process being started, along with that process, we have the capping. Suppose this is the mRNA. So it is going on just actually process. It is going on normally just a transcript. Now at the 5' end, we have the addition of a cap. So that cap is called a 5' cap, also called as RNA cap, or an RNA 7-methylguanosine cap. RNA just 7-methylguanosine cap. RNA 7-methylguanosine cap. And also it is simply described as RNA. This is M7, that is 7-methylguanosine cap. So RNA M7, that is power, and G cap. So these are all the different names for the cap. So the addition of that cap is taking place at the 5' end of the mRNA, the nascent mRNA. We can say there is a newly formed mRNA strand. As soon as what we have, the process of transcription starts immediately. So what is that cap? It is a modified, unusual, that is what is called guanine nucleotide. That is why I said methylguanosine triphosphate. This is entirely different from the normal guanosine or what is called the nucleoside or the nucleotide. So it is a modified unusual guanine nucleotide. Unusual guanine nucleotide, methylguanosine triphosphate. So that has been added, I mentioned already, at the free end, that is at the free 5' end of the nascent mRNA that is being formed. When? Shortly after the start of transcription. So it occurs along with the transcription process. The capping occurs along with the process of transcription. As soon as the transcription has been started, now the capping is also taking place. That is nothing but the addition of, that is, we have 7-methylguanosine triphosphate that is being added. RNA 7-methylguanosine phosphate. What is its importance? So you can see that one just at the 5' end. You know that one transcription always proceeds in 5' 3' direction from the template which is running in 3 days 3' and 5' direction of the DNA. Now, what is the composition of the cap? So the cap consists of a terminal 7-methyl guanosine residue. 7-methyl guanosine residue. And that one is linked to the nascent mRNA, the newly formed mRNA, through 5' triphosphate bond, where as soon as the first nucleotide of mRNA being formed. So it is linked to the first transcribed nucleotide through 5' triphosphate bond. So it consists of, that is simply a terminal 7-methyl guanosine residue. The linkage between the first nucleotide that is transcribed and this component is through 5' triphosphate bond. Now, what is the importance of what is called the capping process? So, with the help of this one, that the mRNA can recognize the ribosomes, or this mRNA can be recognized by the ribosomes. So, only when you have the cap, then only that mRNA can be recognized by the ribosome. Not only that, the RNA is also protected. But the RNA is also protected because of the presence of this cap from the nucleases enzyme responsible for the degradation of mRNA. So this is a protection from the RNA nucleases, the ribonucleic acid, that is enzymes, that is what is called nucleotidases or nucleosidases or simply nucleases. So the RNA nucleases are enzymes, either endonuclease or exonuclease, responsible for the degradation of the mRNA. So the protection is coming from the cap. So the RNA cannot be degraded by the action of what is called the ribonucleases enzyme, and also it is meant for recognition by the ribosomes. So once you have the cap in the mRNA, then only it is being recognized by the ribosome. I mentioned already, cap addition is always coupled with what is called the transcription process. So it occurs co-transcriptionally. So the capping occurs along with the transcription, whereas the tailing occurs after the transcription has been completed. So we have two main events in the second complexity. One is the capping process. Another one, what is called the tailing process. Capping occurs at the 5' free end of the mRNA. The tailing occurs at the 3' free end of what is called, that is mRNA. That occurs after transcription process, whereas the capping occurs along with the transcription. So it occurs co-transcriptionally. Now the tailing process. It is nothing but what is called polyadenylate complex. That is a residue of polyadenyl complex being added. That is why it's called as polyadenylation process, brought up with the enzyme polyadenylate enzyme, that is what we have phosphatase, polyadenylate phosphatase. And now what is happening? The tailing process occurs only after the transcription process. It is nothing but the addition of just adenine nucleotide just many times. That is why it's called polyadenylation process. So it is a covalent linkage. There you have what is called the capping. That cap is linked to the newly formed nucleotide by means of triphosphate bond. But here the tailing process, the mRNA is linked to what is called many adenine, just nucleotide by means of covalent bond. That's why I mentioned here it is a covalent linkage of polyadenyl moiety, the residue, to, that is an mRNA molecule at the 3' end. So in all eukaryotes, you know that one. What will happen? This mRNA has been translated to form the protein. So in all eukaryotes, we have the adenylation process occurs at the 3' end of the mRNA, excepting, that is what is called the histones. The mRNA responsible for the formation, or the mRNA that contains the information for the formation of histone proteins, where there is no adenylation process. That's why I mentioned here in eukaryotes, with the exception of histones. The meaning for that one, those mRNAs which have what we have the message, or which are acting as a template for the formation of histone protein, and there is no polyadenylation process at the 3' end in the case of just the mRNA, which is acting as a template for the formation of histone proteins, excepting the mRNA, which is acting as a template for histone proteins. All other mRNAs have this process of adenylation process at the tail end, at the 3' end, just what we have. This is an exception. Now, what will happen? Once the mRNA has been formed, it has been cleaved from the DNA. Cleaved means that it is being dissociated from DNA after the transcription process has been completed through the action of certain enzymes, what are called the endonucleases. Either exonucleases or endonucleases, we have the enzymes responsible for just actually separating the mRNA or any other RNA molecules or breaking the RNA molecule. Nucleases, digesting or causing the degradation. So once what is called actually the process has been completed, namely the transcription process, the newly formed mRNA has been cleaved from the DNA, which is being dissociated from the DNA by the action of the enzymes, namely endonuclease complex. So after that process, once actually the mRNA has been cleaved from the DNA, what will happen? Nearly about 200-300 adenosine residues. That's why it's called as polyadenylation process. As soon as the transcription has been completed, now the newly formed mRNA has been dissociated or cleaved or separated from the DNA by the endo or exonucleases, the enzymes, and now we have the mRNA, the process of transcription completed. At the 3' end, nearly about around 200 to 300 adenosine residues are added to the 3' end at the cleaved site. So capping occurs at the 5' end, and just we have the tailing occurs at the 3' end. This is being performed by polyadenylate enzyme, phosphatase, or we can say the polymerase enzyme, because we have the formation of polymer, formation of many adenine residues being added to the tail end, that is the 3' end. That is why the enzyme is called polyadenylate, that is polymerase, because many, that is monomers, have been added. Now what is the importance of tail? So this polyadenylate tail is always bound with a protein. Once it is formed, it is being once it is being added, many, that is adenine residues are added to, that is the tail end. It binds with what is called a protein. So this polyadenyl tail along with the protein bound to it normally protect the mRNA from degradation by the action of what is called the exonucleases. These are all the enzymes responsible for just cleaving the nucleotide one after another from the terminal end towards the interior. Whereas the endonuclease is normally breaking the RNA molecule or DNA molecule at any point in the interior, that's why they're called endonucleases. So as the tail is found at the terminal end, and now this one protects the mRNA, which is being degraded because of the activity of exonucleases. So it protects the mRNA from degradation by the activity of exonucleases enzymes, those enzymes acting at the terminal end of what is called the RNA or DNA molecules. And not only that, but also this is what is called the process of tailing, responsible or playing a significant role for transcription, and also the transport of mRNA through the nuclear post to reach the cytoplasm, then translation and termination. So all these events are actually, that is what we have linked together, and all being normally brought about with the help of what is called the tailing process. So tailing process is very important in protecting the mRNA, the one which is being formed, the newly formed mRNA, as soon as it has been transcribed, as soon as it has been spliced, and that is brought about by this tailing process, protecting the mRNA from the action of exonucleases. Not only that, but also it is responsible for termination process, translation process, transcription process, and also the transport and export of what is called the mRNA through the nuclear pores. Now we have studied two complexities. Complexity number one, a clear-cut division of what is called RNA, a clear-cut division of RNA polymerase. And complexity number two, under which we have the splicing process, then tailing process, capping process, and addition of, that is what is called UTR at both ends. And what is its importance? So why we have such complexities in the case of eukaryotes? Now the split gene arrangement what we have in the eukaryotic cells normally represent this arrangement, the eukaryotic gene arrangement, namely the split gene arrangement having exons and introns, and represent probably an ancient character of the gene that was present in the past that has been normally passed on. So the split gene arrangement is one of the important, just what we have, ancient character or the feature of the genome. And secondly, we have the introns. The presence of introns is a reminiscent or the leftover of antiquity. The ancient character has been retained somehow, just like the vestigial organ. So we have number of vestigial organs in the body without actually any functions. Like that, we have the introns being found just as a vestigial organ, a reminiscent, a leftover material of the ancient one, namely antiquity. Now the process of splicing, why we have the splicing process? You know that when splicing is normally brought about by some complexes, namely the spliceosomes, having RNA and protein. So the process of splicing normally represents actually the dominance of the RNA world, as we have many RNAs, and without RNAs there is no process of translation to form specific proteins. That is why the dominance of RNA in genetics play a major role for transmission of information. I mentioned already the DNA is meant for the storage of information, whereas the RNA is meant for actually the transmission of information. So without which we cannot have the process of translation to form protein, etc. So normally in genetics the RNA plays a major role. That is why the process splicing occurs. So the process splicing represents the dominance of the RNA world, and without RNA we don't have the ability of translating actually the message in DNA to form the protein. That is why they play a major role. They are dominant, that is in the genetic field. So these are all the major importance or the significance of complexities. That is the presence of split gene, the presence of introns, the process of splicing, all represent actually the complexities. Why are they just normally found in the case of eukaryotic cells for different meaning? Now we have to compare the process of DNA replication and transcription. So in what way just they are actually similar to each other, in what way they are different from each other. Let's take first one the similarities. Let's compare the similarities. You know that one in both transcription and replication process of DNA. In both the process, normally they copy the DNA strand. In replication, we have the copying of DNA strand. In the case of what is called transcription, we are copying the DNA strand. So there is similarity, both copying DNA strand. So in both the cases, you know that one, the DNA molecule, the two strands of DNA become normally, that's unzipped. The unzipping of the DNA strands and a new RNA strand or a DNA strand being produced. So in both the cases, so the hydrogen bonds between normally the bases or between the two strands have been broken. So we can have the template strand which is responsible for the synthesis of any new strand of DNA or any new strand of RNA. So in both the cases, the two DNA strands of a single molecule get unzipped, and then they are acting as a template to form either RNA or DNA. And both the events happen to occur only in the nucleus, not outside. So we are taking the transcription, though we have translation process normally occurs outside, transcription and replication process occur together or separately just in the nucleus. Now what we are following, so as in the case of DNA replication, the principle of complementarity governs the process of transcription. So one template strand is formed, that is acting as a template. The RNA strand which is being produced is complementary to that of the template strand. And likewise in the case of DNA replication, two strands get separated. Each strand is acting as a template. Though in the case of transcription, only one strand is acting as a template. In DNA replication, both the strands are acting as a template. Each one is capable of synthesizing their own new strand. So in both the cases, what will happen? The principle of complementarity governs the process of transcription as in the case of replication process, except in that. So in place of what is called actually except. For example, normally in the case of DNA replication, adenine forms a pair with, adenine base forms a pair with what is called thymine. But in the case of transcription process, the adenosine now forms base pair with what's called uracil. There is a pairing between adenine and uracil, and there is no pairing between adenine and thymine, because in the case of RNA we don't have thymine. So in the case of DNA replication process, there is a principle of complementarity. In the case of transcription too, we have the principle of complementarity, but one exception. In the case of transcription process, the adenosine forms a base pair with what is called uracil instead of thymine. But in the case of DNA replication, the adenosine always forms a pair with thymine. That is what is happening during just the process of replication. So these are all some of the similarities between replication and transcription process. Now we have to compare once again the replication and transcription process. What are the differences? Now you know that one, it is a copying of DNA, first one. In both we have copying of DNA only. So transcription is a copying of DNA to create mRNA. Though we have both, in both we have copying process, but transcription is concerned with the copying of DNA to form mRNA. Whereas in the case of replication process, it is also copying of DNA, but to create more DNA strands or molecules. So we are producing more copies of DNA. There we are producing what is called more, actually a single copy, not more single copy to begin with. So the copying of DNA to create what is called mRNA strand is transcription. The copying of DNA to produce more, that is copies of DNA, is called replication process. That is one of the differences. And in the case of transcription, now the mRNA is a complementary copy of the template strand. But in the case of replication process, the DNA molecules are exactly a Xerox copy of the DNA molecule, we can say. That's why I say transcription is a complementary copy. We are getting the complementary copy of DNA. And now the replication is the exact copy. When you have a single DNA molecule of two strands, it is being actually produced into two DNA molecules which are exactly similar to that of the parallel strand. That is why we can say, that when the replication process exactly similar. So it is a exact copy of the DNA molecule, and that one is a complementary copy of the DNA molecule. And transcription results in the formation of one single stranded RNA. One single stranded RNA being produced. But whereas actually the replication process, it results in the formation of two double-stranded DNA molecule. From a single double-stranded DNA molecule, two double-stranded DNA molecules are produced. But here, from a single-stranded DNA, the template strand, one single-stranded mRNA being produced. But here you see that a single DNA molecule undergoes replication to form two double-stranded DNA molecules. That is one of the differences. Here only one single molecule with a single stranded. Here two DNA molecules and both are double-stranded. That is another difference. Now the fourth difference would be see that one normally the synthesis of mRNA or the complementary strands of what is called DNA, namely the mRNA, being made by RNA polymerase enzyme. So simply the enzyme involved in the process of transcription to form mRNA is RNA polymerase. But in the case of DNA replication process, the enzyme that is involved to produce copies of DNA is nothing but DNA polymerase. So the enzyme itself differs. So in transcription process, the enzyme required is RNA polymerase. For just actually replication process, the enzyme required is DNA polymerase. And normally, the RNA molecule produced by transcription process is shorter than that of the DNA molecule produced through replication process. Size-wise, the mRNA strand is shorter than that of the DNA molecule produced through replication process. And again, DNA replication is a process of mitosis because we are increasing the number of DNA molecules. And whereas transcription is nothing but a part of the protein synthesis. One part of protein synthesis is transcription. Another part of protein synthesis is translation. So, DNA replication is a process of mitosis, and just the transcription of RNA is a part of protein synthesis, one part of protein synthesis, that is the first part of gene expression, namely the transcription process. So these are all some of the similarities between what is called the transcription and also replication process. What we have the process of transcription in both the eukaryotic and prokaryotic cells. Don't forget that in the case of prokaryotic cells, both the events happen to occur simultaneously, co-transcriptionally, that is a translation. But in the case of eukaryotic cells, first a transcription, then we have a number of processes, namely the processing, and finally we have the translation process, and both occur in two different compartments separately. After transcription only we have the translation process. Again, the capping process, that is one of the events happening during the processing. occurs just normally along with the transcription, co-transcriptionally, whereas the tailing process occurs only after the transcription, that is after transcription only, post-transcription process. Now, one of the events what we have in the examination point of view, what is terminism? And just actually any ism is actually reported by a person with a name. We are adding the word suffix ism like that here. It was reported by Temin in 1963. He observed that even a reverse transcription process in one of the viruses, the one which produces tumor, the one which contains RNA as a genetic material. So in the case of RNA tumor viruses, that one is the ability of synthesizing what is called the complementary DNA, then which code for the synthesis of normal DNA. This is what is happening. See, if the DNA has been synthesized under the direction of RNA, then we can say this is a reverse transcription process. So, the synthesis of DNA by RNA is an example for reverse transcription. And so also, termination is an example for reverse transcription. So, transcription process, DNA directs the synthesis of RNA. In reverse transcription, RNA directs the synthesis of DNA, which occurs in the reverse process by means of reverse transcriptase enzyme, normally found in the case of that one retroviruses. Likewise, here one tumor-inducing viruses, RNA virus, it can synthesize DNA, and that is why it is comparable to the reverse transcription process, namely the terminism. One such question came in the Ames question paper. Now, this is the event what I described in the case of eukaryotic cell, the process of transcription. Now, you see that one, the transcription proceeds. This is what we have, the nascent or newly formed mRNA. I already mentioned the capping process occurs normally at the 5' end along with the transcription. As soon as the start of the transcription, the capping also occurs to just normally for the recognition of this that is mRNA by the ribosome. So the capping, you see that one cap is formed of 5' end, that is it is 7-methyl guanosine triphosphate, 7-methyl guanosine triphosphate, Mg triphosphate, 7-methyl guanosine triphosphate. So this is a capping process. Now this is what we have, the primary transcript having both the introns and exons. Now by means of RNA splicing, the introns have been removed so that we have only the exons. They are all joined together in a defined order. And as soon as transcription has been completed, now the polyadenylation process with the help of polyadenylate polymerase. This is nothing but the addition of what is called many adenine nucleotides. It is also responsible for protecting the mRNA from degradation by the exonucleases. Now this is the fully formed, the matured. This is the primary transcript with both exons and introns. Now the fully matured mRNA having only the exons with the cap at the 5' end and the tail, that is polyadenyl tail. That is why the green part represents the polyadenyl tail at the 3' end. Again the same picture, just somewhat in a detailed manner. We have the transcription unit consists of, that one, the promoter, then actually the gene for coding the RNA, then the terminate. And this is what we call the transcription starting site, and this is the upstream of gene, and this is the downstream of genes, what is in order in the case of bacteria. Now this is a site for the termination of the transcription. Termination, transcription actually being done at that site. This is the starting site and that is the termination site. Now this is what is called the RNA transcript having a cap at this point, and now this red part represents what is called the tail. But here it is represented as a terminated here, but it is normally a tail region after a maturity occurs, after the processing completed. Now it is called the matured RNA or RNA transcript ready for the translation process. Before it is being actually what is called splice, it is called primary transcript or pre-mRNA or unmatured mRNA. Once the processes, namely that is what is called the splicing processes have been completed, then it is called a matured RNA or mRNA, not considered as heterogeneous nuclear RNA. Now the next part, so far we have studied mainly about the transcription process. And the next one, how far the message from the DNA has been transferred to mRNA and then being translated to form protein. And for that you have to know something about what is called genetic code. So what is the genetic code? So we need certain things for simply the process of translation. So the process of translation normally requires the transfer of genetic information, what is contained normally in the DNA in the form of polymer of nucleotides. So the process of normally translation requires the transfer of genetic information from a polymer of nucleotides, namely the DNA, to the polymer of amino acids to form protein. And now this is done. This could be done with the help of what is called genetic code. So the information from the DNA has been transferred to form protein, has been transferred to mRNA, which is being translated to form protein. And for that we need certain genetic code for each amino acid. So that transfer of information normally done by the genetic code. This code normally directs the sequence of amino acids during the process of what is called the protein synthesis. So with the help of genetic code, the one which directs the sequence of amino acids to form what type of amino acid to be added to form a polypeptide protein, that is being decided by the code. And that code is normally coming from the DNA and passed through the mRNA, then to form the protein. That is what we call the central dogma, what is happening. So the DNA undergoes transcription to form what is called mRNA, which undergoes transcription to form protein or polypeptide. So the information from the polymer of nucleotides in DNA is transferred to the protein through what is called the genetic code. It is the one which directs the sequence of amino acids to form proteins during the building of protein or during the process of protein synthesis. Let's see some of the persons actually contributed towards this genetic code, what is called the historical aspect. The first person, George Gamow. You know that one we have only 4 bases in a DNA molecule or in RNA. Adenine, guanine, cytosine, thymine, or in the case of RNA we have adenine, guanine, cytosine and then uracil. And normally in nature we have nearly 20 different amino acids. 20 different amino acids. So if you have nearly 20 different standard amino acids, so possibly the code should constitute or contain nearly about a combination of bases. Not only one base is enough to code for an amino acid because we have 20 amino acids. So we need 20 codons a minimum. So a code is a combination of many, just what are called the bases. So a code being formed by the combination of many bases. He also actually postulated that 3 nucleotides normally forming what is a codon. So 3 nucleotides must be normally employed to form a code or codon, and that will encode 20 standard amino acids by any living cells during the process of protein synthesis. So not 4 bases enough to form just actually encode 20 different amino acids. We need actually a combination of 3 nucleotides that are employed to code for 20 different amino acids by the living cells during the process of protein synthesis. So it is a combination of many bases. That is the meaning for that one what he proposed. As a result, what will happen? A permutation combination of just we have 4 cube, 4 into 4 into 4, that result that generate nearly 64 codons possible. So in a codon we have first letter, second letter and third letter. Like that we have combination of what is called just 64 codons. To specify nearly 20 different amino acids. You know the number of amino acids less 20, but the number of codons you know that will nearly 64. Now the second person, Hargobind Corona, the most important person, artificial synthesis of normal DNA and RNA. So he normally synthesized RNA molecules with well-defined what are called that is combination of bases, adenine, guanine, cytosine and uracil. Just actually from homopolymers and then copolymers. By chemical method, another person by enzymatic method, but here by chemical method, he prepared or he just made RNA molecules. And that is the role by Hargobind Corona. But it was a person by name Marcel Nierenberg. When you are talking about genetic code, you have to remember this person. So he was the first person you know that would elucidate the nature of genetic code, the nature of codon. That is in 1961 in National Institute of Health in America. He was the first person expose or elucidate or explain the nature of codon. in 1961, that is at the NIH. Now what he used? And he used a cell-free system without any cell. He prepared actually without any living medium, he synthesized protein. And also, he just simply translated a sequence of RNA, what is called polyuracil RNA sequence. So he was the first person actually elucidate the nature of a codon. He synthesized protein in a cell-free system, and also he translated polyuracil RNA sequence, and normally deduced that, exposed that, revealed that is the codon U specified the amino acid what is called phenylalanine. So this codon U specified the amino acid phenylalanine was the first codon that was disappeared. So he used cell-free system for the synthesis of protein, and also translated this U polyuracil, that is what is called RNA sequence, and deduced that, actually explained that, revealed that, or disappeared that this is meant for what is called the amino acid phenylalanine. For phenylalanine there are two codons, U and another codon UUC. And the first codon that was disappeared, that was revealed to the public, was this codon named U for phenylalanine. Now the next person, Severo Ochoa. So Nirenberg, Ochoa, Korenberg, all related to the genetic code, but most related person we have Nirenberg. So he made actually RNA molecule by means of enzymatic method by using polynucleotide phosphatase. By using this enzyme, he synthesized RNA molecule in a template independent manner, without any template, without any model. Simply added the nucleotides with the help of enzymes. He joined all these things together to form just namely, that is or simply RNA molecule. And also he translated polyadenine RNA sequence. That person polyuracil RNA sequence, this person polyadenine RNA sequence, namely A. And I explained that the codon, that is A, is meant for the amino acid lysine. It is a codon for lysine. A is a codon for phenylalanine. This is a codon for lysine. So Ochoa just disappeared, that's A, the codon for lysine, and Nirenberg disappeared, the codon U for phenylalanine. And later this, it was Philip, one of the persons exposed the split gene along with Richard Roberts. So Nirenberg and Philip later revealed that the triplet nature of the genetic code, and disappeared the codon of the standard genetic code. So the codon nearly about the triplet nature of the genetic code was explained or exposed by, that is Nirenberg and then Philip. And then just actually Nirenberg along with just Philip, along with just Holy, along with just the person what we said, Korenberg, all contributed towards the genetic code. That is why in 1968 Corona, Holy and Nirenberg were awarded the Nobel Prize. And these two persons, namely Nirenberg and Philip, they were able to determine nearly about 54 codons out of 64 while they did their experiments. So out of the 64 codons, 54 codons were disappeared or exposed or determined by Nirenberg and Philip. For that one he was awarded Nobel Prize. Don't forget, you have to remember the name for genetic code, mainly Nirenberg. Other persons are only after that. Ochoa, that is enzymatic preparation of RNA molecule. And then we have just Hargobind Corona. He is responsible for the synthesis of RNA by chemical method from known bases. Now what is genetic code? So normally we have a relationship between whom? So the genetic code is the relationship between the nucleotide of the DNA and then being transcribed into RNA. So we can say it is a direct relationship. If you are taking the DNA, it is indirect relationship between what is called the DNA nucleotide and then what we have, that is the polypeptide chain amino acid. But genetic code is a direct relationship between the nucleotide sequence of mRNA and the amino acid sequence of a polypeptide chain and protein, indirectly with the DNA, but actually it is a direct relationship between the nucleotide of mRNA and the amino acid sequence of a polypeptide chain or protein that is being synthesized. So what is a codon and just like log and anti-log? We have codon and anticodon. Codon is nothing but a triplet of what is called nucleotides in an mRNA molecule. That is, that one code for an amino acid. That one specifies an amino acid. For example, AUG, this is a codon for methionine. Codon is nothing but a triplet of bases, a triplet of nucleotides, we can say. Normally found in an mRNA molecule, that specifies one amino acid. That specifies one amino acid, a single amino acid. For example, AUG is a codon for methionine. Now, what do you mean by anticodon? We have seen already the structure of tRNA, and which in one of the what we have anticodon form, where you have a set of 3 nucleotides which are normally complementary to the nucleotide basis of codon of mRNA. For example, if you have an RNA AUG, this is the codon in mRNA. The anticodon found in tRNA being, for example, CUAC, because A is always forming a pair with U, and then U is always forming a pair with A, G is always forming a pair with C. So if you know the codon, then you can simply say what is the type of anticodon found in tRNA. For example, if you have a codon triple G, now the anticodon we have triple C. If you have the codon, for example, triple A, the anticodon tRNA would be, for example, U, like that. So it is a triplet of nucleotides found in tRNA, and that is a triplet of nucleotides found in mRNA, and both are complementary to each other. And a codon specifies one amino acid. So this, for example, triple G for glycine and AG for methionine, like that we have. Now, what are the properties of a genetic code? If we are taking the genetic code, the 64 codons, all the 64 codons would be the same in all organisms, starting from amoeba up to human beings. That's why we can say the codons are universal in nature. For example, from prokaryotes like bacteria to human beings. Triple U, this is a codon for phenylalanine. There's no change. But there is some exception we can have. But in nearly 99.9%, we can say 99.99%, in all organisms, we have the same type of codon for a particular amino acid. For example, triple U. This is a codon for phenylalanine, starting from bacteria up to man. I mentioned already there are certain variations in standard genetic code. For example, in the case of mitochondria, we have one codon UGA, coding for the amino acid tryptophan. But the same codon in the case of eukaryotic cells, just in the what is called the nuclear codon, it is considered as a stop codon because it stops or terminates the process of protein synthesis or the transcription process, not protein, it's a transcription process. So EGA is a codon for tryptophan in mitochondria. The same codon just normally found in the nuclear codon, and that specifies what is called simply, that is for a stop codon, the one which stops or terminates the process of transcription. And similarly, in the case of mycoplasma, we have one codon named CUG. This is a codon for serine, but the same codon is for leucine in the case of yeast. So this is a slight variation, 1 or 2 cases only, not in many cases. That's why I said 99.9%, even 99.99%, we have the codon would be the same in all cases. That's why I said it is universal. So these are all only 2 exceptions. UGA for tryptophan and mitochondria. The same one is a nonsense codon or a stop codon or a terminus codon in the nuclear region. That is the nuclear mRNA we can say. So in the case of mycoplasma, you have CUG for serine. The same one is meant for leucine in the case of yeast. Now what is the second character? Only one codon is normally specifies only one amino acid. One codon, one amino acid. That's why I said what is called the codons are always specific and ambiguous. One codon specifies only one amino acid, not more than that. We have many codons for an amino acid, but only one codon specifies one amino acid. That's why I said the codons are unambiguous or specific in nature. In some cases, you know that one, a single amino acid is coded by more than one codon. For example, for phenylalanine, there are 2 codons. For glycine there are 4 codons. For just valine there are 4 codons. And for just serine and arginine there are 6 codons. But there are no 3 or 5 codons for any of the amino acids. So the amino acids have either 1 codon or 2 codons as in the case of phenylalanine, or we have just actually 4 codons as in the case of glycine or valine, or we have 6 codons. There are no amino acids having either 3 codons or 5 codons. But anyway, normally some amino acids have more than 1 codon. That is why we can say the code is a degenerate code. So it is called the process of degeneracy, the phenomenon. Now, normally the genetic codes are ordered, arranged in a specific order. They are ordered manner. Now, out of the 64 codons, only 61 codons are considered as the functional codons. The remaining 3 codons are called the non-functional codons. They do not specify an amino acid. That means they do not have any message for an amino acid. Hence, but they have actually, that is why they are called as nonsense codons, because they do not have any meaning for an amino acid. But they would actually have one function. What function? They stop the process of transcription or terminate the process of transcription, hence called stop codons. This is because they stop or terminate the process of transcription. As they have no meaning, as they do not have any meaning for amino, as they do not specify an amino acid, they are called nonsense codons. So what are the 3 nonsense codons? They have their own specific names: UAA, UAG and UGA. So these are all the 3 nonsense codons: UAE, UAG and UG. Now the UAE codon is also called ochre, and then UAG amber, and the last one opal. Ochre, amber, opal, UAE, UAG, EG. They stop the once these codons are exposed during the process of transcription. Suppose for example there is one codon for amino acid, serine, and that codon just that is the last amino acid, so we have to complete the process of actually transcription. As soon as actually that codon is being reached, now one of these 3 codons being exposed to stop the process of transcription, hence called the stop codon or termination codon. Now one of the specific character of the codon AUG. So this codon has dual functions or double functions. So it is called as the initiator codon because the process of transcription always starts with this codon only. That is number 1. That is why it's called initiator codon. So AEG is an initiator codon. But it also specifies an amino acid, namely methionine. In the case of bacteria, that is formyl methionine. So it has dual function, coding for an amino acid methionine or formyl methionine in the case of bacteria, as well as it is for actually acts as a starting point for the transcription process, hence called initiator codon. And this is the codon which starts or begins or initiates the process of transcription. But in the case of bacteria and archaea, this initiator codon is different. So in the case of bacteria and archaea, you have for example the codons are GUG or UUG. So the last 2 codons will be the same. The last 2 letters will be the same. The first letter has been changed. So in the case of eukaryotic cells, we have AUG is the initiator codon, but in the case of bacteria, it is GUG, and in the case of archaea, it is about UUG. Remember that one. So when you are reading the codon in mRNA, it is always read in a contagious manner. That is without any comma, without any punctuation. It is being written continuously without any comma. That is called contagious method of writing. So the codon is read in an mRNA contagious manner, that is commaless. Always actually there are no punctuations made with the codons. Now here is a table showing the codon. You see that one 64 codons, and they are specifying different types of amino acids. So some of the amino acids are that one non-polar. For example, this yellow color represents the amino acids of non-polar type. So phenylalanine, leucine, just isoleucine, methionine, just we have that is valine, and we have for example glycine, alanine, and then just a protein, all are non-polar type. And we have the polar type. These are all the polar types: serine, threonine, etc. Then some of the amino acids are basic amino acids. Now these are all the basic amino acids: lysine, arginine, histidine. So one of the properties of that when the nucleosome, in the case of actually we have the histone proteins. The histone protein is a basic protein, that one. which contains large amount of what is called histidine lysine. That is why normally you know that one. The DNA, which is a negative molecule, is always bound with the basic protein having a positive charge. This is because of the presence of these, actually the basic amino acids. And we too have the acidic amino acids, for example, aspartic amino acid, glutamic acid, all these things are examples. So anyway, this is a table showing. So also other tables, we have the 3 stop codons. Now this is given clearly. Now these are all the stop codons. And this one, methionine, is the starting or initiated codon. The glycine, actually the simplest amino acid, having, you see that one, 4 codons. Valine to have 4 codons. We have just 2 amino acids have only 1 codon. For example, the amino acid tryptophan, the amino acid methionine. These 2 amino acids have only 1 codon. UGG codon for the tryptophan, AG codon for the methionine. And we have 2 codons, for example, phenylalanine. And then I mentioned there are no 3 codons for any of the amino acids. For 4 codons, we have valine, glycine. And there are no amino acids having 5 codons, but we have amino acids with the 6 codons, say, for example, serine 4 plus 2. Then arginine 4 plus 2. So 6 codons for that. So like that. So go through that one. The next one is also the same one with some. Actually, the first letter, the symbol. So this is a symbol for phenylalanine, F. And then using like that, we have the symbol which is being given, W for tryptophan, R for arginine, S for serine, G for glycine. So these are a symbol used for different types of amino acids. The same one, and that may also be given in compressed manner. For example, if you are having GCN, that is actually alanine with the 4 codons. So GCN, actually the compressed form of the codon, and which includes 4 codons. Arginine, we have just the CGN, MGR, which include nearly 6 codons. Like that, go through. It's not necessary, hard to go through simply. Now this is an illustration of a single stranded mRNA having nearly 7 codons. This is codon number 1, always the first codon A with G. Initiated codon, and always the transcription starts with this process, namely that is a protein synthesis or the transcription, where you have the AG is the initiated codon which begins the process of transcription. That is why the first amino acid in any protein is methionine, which is being replaced in some cases, may not be replaced in some cases. Not all proteins have methionine as the first amino acid. Once formed, it may be replaced during the configuration process or conformation process. Now this is the problem. You see that when predict the sequence of amino acids from the sequence of nucleotides in mRNA. Now these are all the sequence of nucleotides in mRNA. And now you have to just find out. I mentioned already the first amino acid, always methionine, because the initiated codon is AG. So these are all the amino acids. So in the first problem, now the nucleotide sequences are given, you have to find out the amino acids. The second problem, just actually the amino acid sequences are given in a protein, you have to find out what is the type of a codon for each one. This is what we have some problem, you go through that. Now mutation and genetic code. So normally the relationship between the genes and DNA are best understood with the help of what is called the point mutation. The mutation that occurs at the gene level or at the base level or at the nucleotide level is called what is known as the point mutation. The rate of mutation is normally very slow because the DNA or the gene is a very stabilizing molecule. The error or the mutation occurs, the rate of mutation is about actually 10 to 100 million bases. One error in 10 to 100 million bases. This is because of what is in order during the replication process of DNA, the proofreading ability of what is called the DNA polymerase. While the DNA being replicated, is there any correction that is being made properly by the DNA polymerase, that is called the proofreading. So because of the proofreading ability of DNA, so what would happen, the rate of mutation is very slow or less. One mutation per just actually 10 to 100 million bases. Out of this 10 to 100 million bases, just only one mutation or one error occurs. This is because of the proofreading ability of a DNA polymerase. So we have different types of gene mutations. The first type what we have with reference to, so altogether 4 different types of mutations in our syllabus. One is a missense mutation. second one silent mutation, nonsense mutation, and also the last one frame shift mutations. So what do you mean by missense mutation? It is nothing but the substitution of a base or an amino acid. Because of the substitution of a base, an amino acid has been substituted with the protein molecule. So that is why I mentioned here it is a type of point mutation that causes an amino acid or base substitution. So it is an example for substitution mutation. As a base has been substituted, immediately what will happen in the protein, an amino acid has also been substituted. The best example for this point mutation or the missense mutation is sickle cell anemia. Now in the case of sickle cell anemia, you know that a normal hemoglobin contains 4 polypeptide chains, 2 alpha and 2 beta. In the beta globin chain, in the 6th position, we have just what is called actually amino acid, glutamic acid. Glutamic acid. We have seen already discussed this one, glutamic acid in peptide number 6 of beta globin chain. So it is coded by GUG. This is a codon. This is a codon. That is what we have. The codon. This is a codon. GAG is a codon for this amino acid. As a result of single base substitution, what will happen? The A has been substituted by U. What will happen? The amino acid has also been substituted. Just the glutamic acid has been replaced by valine. So out of the 586 amino acids, the point mutation occurs at only one point. This is nothing but a simple substitution of a single base. A has been substituted by U. Ultimately, in the protein molecule, the glutamic acid in peptide number 6 of beta chain has been substituted by valine. That results in a disorder. So out of 586, only one amino acid has been replaced. Because of actually one base has been replaced or substituted. This is an example for missense mutation. Then, silent mutation. So normally I mentioned already, for example, glycine. There are 4 codons. These are all the 4 codons. GGA.
SMS from a node sender.
That is triple G. These are the 4 codons. This is what is called redundancy. This is called redundancy. That is 2 or more codons for an amino acid. That is called what is called redundancy. And because of that one, what will happen? Some point mutation. Suppose let us take this is a normal codon for glycine. It undergone mutation so that this A has been replaced. So we have, for example, triple G. So mutation occurred, but that mutation is not expressed because we have all the codons, all the 4 codons. Even after mutation, the codon can specify the same amino acid so that there is no change resulted in the protein molecule. So when a codon has been normally mutated, the resultant codon, if it is not specifying the same amino acid, what actually the mutated codon produce, there is no change that is in the protein sequence, namely the amino acid sequence of protein. So there is no external visible appearance because of that mutation, because the resultant mutant codon also specify the same amino acid. And that is why what will happen, an error that would not, so you see we have another definition what I given here, an error that would not affect the protein, because the hydrophobicity or hydrophilicity of what is called the protein is maintained because of the equivalent substitution of amino acids. The same amino acid has been substituted. There is no change with the reference to what is called the hydrophilicity or hydrophobicity of the protein. It is being maintained as such because some of the amino acids are hydrophobic, some of the amino acids are hydrophilic, and that hydrophobicity, hydrophilicity of that particular protein has been maintained because of the equivalent distribution or equivalent substitution of a amino acid. Like this, when this GGA undergone mutation to form triple G, and that codon, this codon both normally specify the same amino acid. That is why there is no visible external appearance. Okay, so these are all the 2 types of mutations, and we have 2 more, namely just the silent mutation. After that we have nonsense mutation and frame shift mutation. I will continue the same tomorrow, just about the remaining and also the process of translation under this molecular basis activities. Okay, thank you. Goodbye.